Okay, hi everyone. Thank you so much for joining in. I'm Priya from the CAC department from the Indian Institute of Technology, Kanpur. I would like to thank ISMB for giving me an opportunity to present my work Falcon, which is a single cell level mutation detection algorithm for high throughput panel sequencing datasets. Let's get into a little bit of detail on why do we need to infer somatic mutations. Somatic mutations are acquired genomic variants that occur over the lifetime of an individual and they're not passed to the offsprings. And these are the most common cause of cancer. Cancer is an evolutionary process that occurs due to acquired somatic mutations over a period of time. And we need to infer this tumoral progression in order to provide better therapies to cancer patients. Somatic mutations may include structural variants, which may end up affecting a certain part of the genome, for example, insertion, deletion, or copy number alteration. Now, most occurring somatic mutations in cancer are single nucleotide variants. So, single nucleotide variant simply is genomic alteration at a single genomic locus. For example, in this case, the base A changes to base C. This is an example of a single nucleotide variation. And these are the most prevalently occurring genetic alterations in cancer. And we need to infer SNVs at the level of the resolution of single cell in order to understand the tumor better. So, in order to achieve this, the emergence of single cell sequencing technologies have helped us. The single cell profiling methods have enabled us to study the progression of cancer at the level of a single cell. We are now able to resolve the heterogeneity that lies within the tumor. So, earlier we used
mixture of signals coming from several cells. But now we are able at the level of single cell where we are able to find
the genetic markup, the genetic information coming from every single cell. Due to this, many downstream analysis have been have been made better. For example, clonal lineage inference. For example, inference of the clonal substructure. We are also able to infer better tumor evolution with the advent of single cell sequencing technologies. Nonetheless, it comes with its own sets of technological limitations. For example, uneven coverage, for example, susceptibility to false negatives due to allelic dropout. So it may happen that we have a true single nucleotide variation present at some genomic locus, but we don't have any alternate reads to support that. So we may end up calling it a non-mutation when actually it was a mutation. And the third case is a false positive error, where due to erroneous amplification, due to the infidelity of the polymerase, it may end up resulting into the amplification of a wrong nucleotide. And we may end up calling a positive mutation at a place where there was no mutation at all. On top of the existing technological limitations that are associated with the single cell sequencing technologies, there are now new pipelines that have been emerging and they are able to produce high throughput sequencing dataset. These are panel sequencing methods which do not take into consideration the whole genome, but rather a panel or a targeted like amplicon, and they amplify the disease specific genes, and due to this they are able to parallelly sequence thousands of cells. And the existing variant callers are not able to scale up for such parallel sequencing involving such high throughput dataset. These are two recent studies which have used such pipeline in order to analyze the data, and they have sequenced over 5,000 to over 12,000 cells. And the current single cell variant callers are only applicable or only work till almost hundreds cells at max. So there is a need of a new variant caller which can scale up for such high throughput dataset. So we have designed Falcon, which is a statistical phylogeny-aware variant calling algorithm for such high throughput dataset. Falcon starts out with the read count matrices that are obtained as an output to the pipeline that I just described in the last slide. So we start with alternate read depths, total read depths. We apply a 5-step filtering process in order to find the putative variant sites. And we, on top of that, we calculate the mutation likelihood for all such putative sites and every cell based on some sort of beta binomial distributions that I will explain later. Using these mutation likelihood matrices, we cluster the single cells. Now, after we have found the clonal structure, the clonal substructure of the tumor, we initialize a random clonal tree on the same, and we make it go through an exhaustive tree search in the phylogenetic space, and the maximum likelihood tree is chosen from there. based on the branch, like mutation placement on the branches and the phylogenetic reconfiguration of the trees. Using this tree, we perform a clonal level variant calling. So you can see it is somewhat like this, that if this green mutation is at the root, which basically means that all the clones have harbored this mutation. So for the green mutation, all the clones will be genotyped as 1. Now, based on the cluster association of every single cell, we perform the single cell variant calling after this. This is a basic overview of Falcon. Now let's dive into the details of how do we calculate the mutation likelihood matrix, which is the most important. In order to calculate the mutation likelihood matrix, we use 3 different events that can occur. So, which is the genotype configuration. The alternate depth, allele depth is taken care of by the parameter P, and the total read depth is taken care of by the parameter Q. Let's take the first case, which is the homozygous reference. So in this case, we are not finding any alternate evidence. So that basically is the case of no mutation at all, which is G equals 0. And we are empirically finding that a beta binomial distribution is able to capture this better. So we are modeling a homozygous reference case using a beta binomial distribution with parameters alpha q and beta q. Now the next case is the heterozygous mutation, in which we are finding reference evidence as well as alternate evidence, and this is a little bit tedious to configure. Empirically, it has been found that the alternate, so these are empirical values, the gray bars, and it is found that the classic binomial distribution is not able to capture the heterogeneous sites well. It is not a single beta binomial distribution is also not able to capture this well, but rather a mixture of 2 symmetric beta binomial distributions is able to capture this alternate nucleotide distribution better. So we are also using 2 beta binomial distributions which are scaled WQ, which is the allelic dropout parameter, and these parameters are actually can be learned with respect to the data, which I will explain in the next slide. So this is how we have modeled the heterozygous case, which is G equals 1. And now the third case is G equals 2, where we are not finding any reference evidence. So in that case, only what we have done is we have used the same beta binomial distribution that we used in the first case, but this case is completely different to the first one. So we have just swapped the alpha q and beta q parameters. Now these parameters can be these parameters of the beta binomial scale up linearly as the read depth increases. This has been motivated by the papers that are stated below. So you see, according to the total read depth, the value of this is allelic dropout parameter, and these two are the parameters for the symmetric beta binomial distributions. With increasing total read depth, we are seeing that W, alpha 1, alpha 2, all of them are increasing. So we have learned this for our method as well. After calculating the genotype likelihood, we first calculate the mutation likelihood. How do we do that? This LG equals 0 is the genotype configuration where there was no mutation. We normalize this and we subtract it from 1. This makes Lij the likelihood of a mutation occurring at a site I and cell J. Now let Lij be the mutation matrix. This is something how the mutation likelihood matrix looks like. For all the cells, there will be values. These are the sites. These are the putative variant sites. It will be value that will range between 0 to 1. Now, based on, so you can think of these mutation likelihoods as the features for every individual cell. And based on that, you can construct an neighborhood-based affinity graph. And you may perform a graph-based clustering algorithm on the same. Mostly we have used spectral. And using such clustering, we are obtaining like this rich subclonal like landscape for the tumor. How we have we also have used a tree like we are modeling the following events on the tree under the finite sites model. We have used the tree in order to call the mutations. So the 3 events that we are modeling on the tree are: number 1, the heterozygous mutation, where if a mutation occurs at a branch, it goes down the phylogeny. So in this case, the green mutation is harbored by clone 1 and clone 2. The circles in between these clones are the cells, which are clustered using the mutation likelihood matrix, which I just described. The second case we have considered is the parallel case, where like subsequently 2 mutations can occur at parallel branches. So this orange mutation, it will be harbored by clone 1, clone 2, and clone 3. We have also made sure that we take into consideration the loss mutation. That is, if a mutation occurs at some branch at the phylogeny, it may get lost later on. So in this case, this purple mutation is placed at this branch, but it later gets lost on the phylogeny. So the only clone that is going to harbor this mutation is the clone 1. This mutation placement is handled by the vector sigma, and so what we do is we place different mutations at different branches on different tree reconfigurations, and what we do is we maximize the total likelihood of the tree. This is the term for the total likelihood of the tree. This consists of two terms, which is the log likelihood and the regularizer. So log likelihood is pretty simple. I will explain it with respect to an illustration. So these are cells, these small circles are cells. I'm saying if there is a cell that is below this mutation, then I multiply it with the log likelihood of the mutation occurring. And if there is a cell that is below a point where there is no mutation, in that case, this activator function, this indicator function gets active. So we multiply that with the log likelihood of no mutation occurring. So this is a simple log likelihood that we have used. Now we also did not, we knew that we are modeling loss and parallel cases as well, but they do not frequently occur on the phylogeny. So we wanted to regularize that using this regularizer. So this is if there is a parallel mutation occurring, then we make this value, the log likelihood value, more negative using these two regularizers. Now, the best mutation placement and the best tree is chosen according to the argmax function, which will maximize the total log likelihood of the tree. These are some results on the simulated data. We have benchmarked the PHALCON against against some recent state-of-the-art single-cell level variant calling algorithms such as Cypher and Phylogeny-aware. This is done on 2000 cells with increasing number of clones. And we also wanted to test it against another single cell variant caller, which is Sieve, but Sieve was not able to produce any results on such high throughput dataset even after 72 hours. The average improvement of PHALCON over the existing methods is round about in the range of 7.6 to 42% in terms of genotyping accuracy. We also wanted to test whether PHALCON can scale up to 5,000 or 10,000 cells, and unfortunately, these variant callers Cypher and Phylogeny as well failed for over 5,000 cells. So there is a little cross there. But PHALCON is showing consistent performance across all the number of cells. And Sieve again, of course, did not produce any results for the same. Now we also wanted to infer whether we are inferring a good tumor phylogeny as well. So for that, we have used a triplet-based similarity for checking the correctness of the inferred tumor history. And we also included another method, which is COMPASS, which, if given the true set of single nucleotide variations, gives you like a reconstructed phylogeny of the same. And PHALCON is outperforming all the methods by large margins, even in case of inferring the tumoral phylogeny. This is another strong point of PHALCON, which is the runtime improvement. On y-axis, you can see the time taken, and we have compared it against the methods. This testing is done on 2000 cells on 5 datasets. And you are seeing that Cypher almost takes around 18 hours, Phylogeny almost takes around 16 hours, but PHALCON is able to complete genotyping or variant calling on datasets consisting of 2000 cells in as less as 22 minutes. And of course, Sieve was not able to produce anything after 72 hours as well. We also wanted to test PHALCON's runtime with increasing number of cells, and we took it till 10,000. And even on 10,000 cell, it was taking around about 2 hours and not more than that. We also tested PHALCON on a real-world dataset as well, 2 datasets. The first one is TNBC, which is triple negative breast cancer, mostly known to be the most heterogeneous type of cancer. And we are able to orthogonally validate the somatic mutations that are inferred by PHALCON using the bulk sequencing data as well. And you are seeing that majority of the sites that are inferred by PHALCON are also orthogonally validated by the bulk data. We are also detecting clonal mutation in TROC-R in all 5 patients. And these are, so the CAD score for this variant is 25.3. CAD score basically tells the deleteriousness of the mutation. And this high CAD score simply means that the mutation is deleterious. We also checked it against MutePred2. So this threshold of 0.5 tells that this is a pathogenic mutation, and it's a deleterious mutation. In 2 patients, PHALCON also identified subclonal mutations in RASL2 gene. So we are inferring somatic mutation in the RAS gap, which is important. And we also found some literature going on that is telling that RASL2 is promoting triple negative breast cancer progression, and mutations in the same may cause deleterious effects. PHALCON is able to resolve intertumor heterogeneity in triple negative breast cancer. We are seeing that we are inferring a good clonal substructure, and we are also inferring clones with as less as 2 cells as well. So basically, that is a testament of that we are able to detect the rare subclones as well. Yeah. So and we are also able to infer a rich phylogeny. We are also detecting somatic mutations in many important genes, and they have a high functional impact. That is as it is visible from this diagram. Next, we tested PHALCON on a cohort of 102 samples from acute myeloid leukemia patients. And this is the box plot for the percentage of sites that were validated by the bulk. And we are seeing that over 70 to 80% of the sites of the somatic mutations that were inferred by PHALCON was also orthogonally validated by the bulk data. We also are finding mutation in two very important genes, ASXL1 and U2AF1, in about 96 to 99% of the samples. And this was not very much highlighted from the study we took this data from. And we are currently doing some literature survey on the same, and we are finding that mutations of U2AF1 and ASXL1 both are correlated with poor prognosis, where you can see that the mutant type of U2AF1 and the mutant type of the ASXL1 are overall having low survival probability as compared to the wild type. So in order to summarize, we have created a new single cell level variant calling algorithm, which is able to scale up for high throughput sequencing datasets. The main idea behind PHALCON is that we infer the underlying cellular subpopulation and we perform a clonal level variant calling followed by single cell level variant calling. PHALCON is outperforming all the existing state of the art methods in terms of variant calling accuracy as well as runtime. And we are detecting somatic novel mutations in important tumor suppressor genes which have high functional impact and orthogonal support in bulk datasets. I would like to thank ISMB for allowing me the travel grant, which unfortunately I was not able to utilize. I would like to thank the funding agency PMRF for funding my research and to my Cosmic Lab members. Thank you so much.
Thank you, Priya, for an excellent talk. Do we have any questions from the audience?
Hi, thank you for a really interesting talk. I was wondering if you could elaborate a bit upon the thinking behind your workflow, because what number of mutations are you able to scale this for? And also, how robust is your variant calling inference to getting the wrong clone tree? Because if you have too low of a coverage and you cluster inaccurately, then how much does this affect your variant calling? Thank you.
Okay, so we have used, so coming to the first question, I think the idea behind this is that we want to infer the genealogical relationship between the cells. We are saying that if like for such a high throughput sequencing data sets around 2000 cells, we will not see very much genetic difference, the difference in the makeup of every single cell is not going to be very different from the other cell if they are belonging to the same cluster. Now we have done one thing, we have calculated the mutation likelihood. Okay, so that mutation likelihood is going to make sure that similar mutation profile cells fall under the same clone. When you asked about the number of somatic mutations that we're inferring, we have benchmarked against 50 to 100 somatic mutations. And what was the other question?
Sorry, also about coverage, because if you get an inaccurate clone tree because you weren't able to cluster correctly, how much does this affect variant calling?
So so actually, in order to maintain the quality of the genotyping, we have removed some sites which have low coverage. We can think of something in order to incorporate that. But I would say that because based on the mutation likelihood, we are performing clustering based on that, and we are performing a graph-based clustering. So the cells which will have the same mutation profile are going to belong to the same cluster using the clustering methods that we have applied.
Yes, but just something to think about is that if you have too low coverage, you might not see the differences between cells. So what you think belongs to the cell might not do so. Just something to think about. Thank you so much.
Yes. Yeah.
Thank you so much, Priya. Fantastic talk and fantastic tool. Could you please point us out to the repository of the tool? So because PHALCON is such a popular name, and what jumps out is some PHP framework or something like that.
What I could not understand.
Just could you please show us where is the GitHub repository of the tools? Where can
Oh, actually, the paper will be out soon. Please do give it a read. Actually, we have not put the paper yet on bioRxiv, so all the GitHub and everything will be available there.
Okay, thank you.
So I also have one question. So your model assumes that there's copy number neutrality, right? So you don't consider any copy number changes in the genes, right?
So basically, we have benchmarked against copy number as well, and we are seeing that yes, with increasing copy number, there is a little bit of degradation, but it's still better than the other methods.
Okay, thank you. And last question. So do you have any way of quantifying the uncertainty? So you're always giving basically one tree, right?
Right. Quantifying the uncertainty of the mutations that we are inferring, right?
Yeah, and of the phylogeny also.
Of the phylogeny as well. So we are coming up with a statistical test. Like that is the only reason why this paper is not out yet. We are currently configuring a statistical test that will be based on there's like the same inferring the false positive, false negative, and whatever there is. So we are coming up with a statistical test in order to tell about the confidence of these somatic mutations.
Thank you very much. Let's thank Priya again, all speakers of the morning session. And we now have a coffee break. And reconvene at... Oh, sorry, there's actually one more question.
Oh.
Yeah, thanks a lot.
Thank God I didn't leave.
Do you hear me?
Yeah.
Perfect. Just a naive question on the genotype likelihood. Would it make sense to know use the information, whether it's a transition, a transversion, a CPG, all of these things? Like, is it common to do that, or would that make sense or not?
Well, I've not actually thought about it, but maybe it will be better to have like more information on the same, because that will be able to make our model infer the genotype likelihood even better. The likelihood will be made better if more information is included. So yes, thank you so much for this question, actually.
Thanks.